In this video, you will see how to concentrate your protein solution using the Thermo Pierce protein concentrator for sample volumes up to 6 mL. Do you want to desalt or buffer exchange your protein too? Not a problem. The Pierce protein concentrators will simultaneously concentrate your protein while removing salts, allowing for easy transfer to a new buffer. These disposable devices are conveniently used in a benchtop centrifuge. They contain polyethersulfone, or PES, membrane in a variety of molecular weight cutoffs, including 3K, 10K, 30K, and 100K. The cutoffs are etched on the side of each device for easy identification. Membrane selection will depend on the size of your protein. PES membrane will retain proteins and other molecules with a molecular weight that is at least 2-fold higher than the molecular weight cutoff. The concentrated sample is referred to as the retentate. The molecules and contaminants in solution that are small enough to pass through the pores of the membrane are collected in the filtrate. To concentrate your protein or proteins of interest, place between 2 mL and 6 mL of sample into the sample chamber and cap. Place the concentrator into a swinging bucket rotor, as shown here, or a fixed angle rotor that accommodates 15 mL conical tubes. Make sure that the concentrator has a proper counterbalance. Centrifuge up to 4,000 times G until your desired concentration factor is achieved. The dead stop volume is 30 microliters. The retentate volume can be visually monitored through a clear window with graduations on the side of each device. Use a pipette tip to gently remove retentate from the sample chamber. Up to a 30-fold concentration can be obtained in as little as 15 minutes with protein solutions of 0.1 mg per ml or higher. That said, concentration times may vary significantly based on molecular weight cutoff and sample concentration or viscosity. Typical protein recovery is greater than 90%. If you need to desalt and or buffer exchange your concentrated sample, simply decant the filtrate, reassemble the unit, dilute the retentate to the original volume with exchange buffer, and then cap and mix. Repeat centrifugation followed by addition of exchange buffer until desired solute removal has been achieved. Typically, salt content can be reduced by at least 98% with just one exchange with water or salt-free buffer. Find out more at thermofisher.com/concentrators.